Hi everyone, and welcome to reproductive system. We're going to start with the male reproductive system. So here's the big overview. We've already kind of seen one of these sagittal sections before. But basically when we're going to talk about the gonads, the ducts, the glands, and all these supporting structures. Right? You can see here definition. When we say semen, what we're talking about is the fluid that's going to contain sperm plus all the other glandular secretions. From this sagittal section, we can see the male bladder. Here's the testes external to the body, right? Here's the pubic symphysis, so that's that bone. So anything kind of like here is external. This is all internal, if that's not readily obvious to you. So what we will see is that, right, the testes are going to be what is producing the sperm. They will be stored in the structure called the epididymis. Upon ejaculation, they will travel up this tube called the ductus deferens, or it's also called the vas deferens. Either term is correct. In over that pubic symphysis, past and behind the bladder, where it will join up with secretions from the seminal vesicle. It'll travel through the prostate. The part of the urethra that's coming from the bladder will actually be closed so that urine cannot mix with sperm. And then it will go through the penis and then out the body. So just a quick tour and then we'll get into some of the specific organs here momentarily. So we're going to have a lot of terms that, I don't know, I think in everyday conversation, I don't know if they come up in everyday conversation, but in everyday speak, we may think of them or say them interchangeably, but there is a difference. So the testis itself is the structure that is producing sperm. The scrotum is going to include the skin, the muscles, blood vessels, lymphatic vessels, nerves, and everything else. That is all of that is going to be the scrotum itself. So just a little bit of a difference. The importance here and the reasons why it is external to the body, unlike in females where the ovaries are internal, or even in some animals, is because the sperm cells in humans require a lower than body temperature to survive. If they were inside the body, they would actually die. So that's why they are outside in the testes, wrapped in this scrotum. The scrotum does have a muscle around it called the cremaster muscle. And what that will do is it will either contract and elevate the testes closer to the body, maintaining a warmth if it is cold outside, or it'll relax, lowering them, keeping them away from a hot body, keeping them cool. The testes themselves are surrounded by a white capsule called the tunica albuginea. So tunic just means like a wrapping, and albuginea is like a white wrapping, this white connective tissue. There are all of these dividing sort of septa, these connective tissue dividers that end up forming many compartments. You see this a couple hundred compartments. These are called lobules. Each one of those lobules is going to be filled with 2 or 3 tubules. Those tubules are called seminiferous tubules because that is where the sperm are being produced. So if we take a look at this testis, and we're going to take a look at one of these seminal vesicles, seminiferous tubules, not seminal vesicles, seminiferous tubule, and we take a cross-section of that. This is what we will see. You'll see this in lab as well. So along the very outside, you'll see this is one tubule, that's the basement membrane. We are going to have these spermatogonia along the very outside. These are going to be the stem cells or the dividing cells. Then there'll be these much larger Sertoli cells. These are also called sustenacular cells. They're also called nurse cells because they are going to help nurse or nourish the dividing and developing cells. So it's the spermatocytes, the spermatids, and the spermatozoa. These are all basically the same thing. It just depends on where you are in its development. The sperm cell gets a slightly different name. So not only do they nourish the developing sperm cell, but they're also going to help modulate the effects of testosterone and follicle stimulating hormone on the process of sperm development. What's going to happen is they also phagocytize extra cytoplasm, because basically if you look here, the spermatogonia, again, these are the dividing cells. They have your typical sort of cuboidal shape. Well, it's got to go from your typical cuboidal cell shape to something that looks kind of like a football with a long tail. And that's going to take some, I'm going to say, carving away at the cell, at the cytoplasm to get that shape. And that's what these nurse cells will do. They basically prevent the cells from moving. So as the cell is developing, you do not want it moving just yet. You want to inhibit its movements because these cells don't have a lot of energy. It's not like they have fat reserves or anything like that. So you want them to sort of be somewhat dormant in activity until ejaculation. And that's really when you want them to be activated so that they can make sort of like this burst of a swim toward a potential egg to fertilize. And that's what that hormone inhibin does. It's going to inhibit the movement of these sperm cells. So as the sperm cells are developing, basically, again, these are the dividing cells here along the very edge of the tube. They are going to divide, and as they mature, they're going to go closer and closer toward the lumen of that tube. As they move closer to this lumen of the tube, these nurse cells are also producing a fluid. So again, those sperm cells are being nourished, and that fluid is just simply going to carry them away. Think of like, I don't know, tubing down a river kind of thing, maybe, toward the epididymis, where they're going to be stored until they're mature. So another name for a sperm cell is simply spermatogonium. These are the cells, well, let me take it back. So these spermatogonia are the cells that are going to produce sperm cells. So again, here are the spermatogonia along the very outside. Eventually they will form the sperm cells here in the middle. There are also going to be these, I don't know how well you see this here in this image, but there are these interstitial cells outside of the tubes, and these are secreting testosterone. So that's the source of testosterone in the testes. It's not in the seminal vesicle themselves, but these cells just adjacent to it. So again, these spermatogonia are the stem cells. They are 2N. Now we have to get down to a single N because remember, we're going to get half our chromosomes from mom, half of them from dad. So the spermatogonia are going to divide, and so where they divide into 2N. So you have, at that point, from spermatogonium, it goes to a spermatocyte, where it has like a little tail. It becomes a spermatid. And then when it's pretty much like a fully developed sperm cell, it's then called a sperm cell. So that's what I mean. These terms, spermatocyte, spermatid, sperm cell, essentially just tell you where in its development it is. And the one thing I like about this image, you can see these nurse cells are sort of these really weird large cells that are helping. So here's this dividing spermatogonia that will then help develop this, again, kind of, well, in this case it's circular looking in this drawing, but sort of this cuboidal shaped cell from this and turn it into something that looks like that. So here are just some definitions. So spermiogenesis is just the maturation of the spermatids into sperm cells. So again, these are not mature. These will be the fully formed sperm cells, although again, they are not active yet. And then this term spermiation is where they are released from the Sertoli cells. And again, the fluid produced by the Sertoli cells will push sperm along their way toward the epididymis, where they're going to be stored. Again, no swimming just yet. As far as their shape goes, there are there's three general areas. There's a head, there's a neck, and then a tail. The head will have something called an acrosome. That is going to be really important, and that's also where the DNA is. That acrosome has enzymes that are going to help digest its way to the cell membrane of the egg so that it can fertilize it. The tail contains the neck and the midpiece that will have ATP and mitochondria to produce energy for the tail to move. They don't live very long. They live about 48 hours in the female reproductive tract. And then if you really would like to see sperm swimming underneath the microscope, you're welcome to click on and watch that video. Okay, so let's talk a little bit about our friend's gonadotropin-releasing hormone, the anterior pituitary and follicle-stimulating hormone, and luteinizing hormone. So just as a throwback to our endocrine system, remember gonadotropin-releasing hormone will cause the release of follicle-stimulating hormone and luteinizing hormone from the anterior pituitary. Luteinizing hormone is going to stimulate testosterone secretion, and again, those Leydig cells, or those interstitial cells, another term for those, will secrete testosterone. That testosterone, some of that is going to get converted into dihydrotestosterone that we saw in our endocrine conversation as well. And again, that's what's going to help give that male pattern of birth development and the proper development of male reproductive organs. Testosterone also, along with follicle-stimulating hormone, is going to stimulate spermatogenesis, or the production of sperm cells. And again, this is there will be a negative feedback loop where high levels will then feed back to decrease these levels, so that again we maintain homeostasis. Okay, so sperm cells, as they are being produced in these seminiferous tubules, they then are going to be taken toward these little straight tubules that you see here into this, basically this tubule net-like section called the rete testis. From there, they will go into the ductus, the ductus epididymis, which will take them into and they're stored here in the epididymis. And then from there, upon ejaculation, they will be taken through the ductus deferens up again into the body around the bladder, and then into the urethra. So let's just talk a little bit about the epididymis. That's this structure right here. It's sitting on top of the testes and kind of wrapping around posteriorly to the testes. You'll notice it has a head, a body, and then a tail. Not unlike those are sort of like very typical portions again to organs. Many of these ducts become a single duct, very long. I mean, it's impressive, 20 feet if it's uncoiled. I don't know how they figured this out. That had to be someone's job at some point. Like, I don't know if I'm a little scared or impressed, but anyway, so it's 20 feet if uncoiled, roughly. And again, the main role here is that this is going to be a site of sperm maturation. The movement of sperm is going to increase over a couple-week period. They are not stored indefinitely, so if there is no ejaculation within about a month to 2 month period, they will be broken down. There is smooth muscle in the epididymis, so upon ejaculation, some of the sperm that are being stored there will be sent forward, again, up the vas deferens, in through the body, behind the bladder, where it meets up with the seminal vesicle, in the by the prostate, where it enters into the urethra. As far as what's in the spermatic cord, so again, the spermatic cord will be part of that scrotum. So these are all arteries, veins, nerves, lymph vessels. Again, the vas deferens, along with that cremaster muscle that will help raise and lower it. As far as the urethra goes, I've mentioned this now a couple of times. So as the vas deferens, or ductus deferens, either term is fine, go into the body, around the back of the bladder, joins up with the seminal vesicle here in the prostate. This portion of the urethra right here that's still in the prostate is simply the prostatic urethra. There's this little portion, I don't know, can you see it right in there? It's past the prostate, but it's not yet into this blue penile urethra portion. It's in between. It's in this membrane area. That's the membranous urethra. And then it goes into the penis, where it's the penile urethra. Right? So where the ductus deferens joins up with the seminal vesicle, that is called the ejaculatory duct. The seminal vesicle will release its secretions just before it releases semen, and we'll talk a little bit about what these secretions do. So specifically, we'll talk about the seminal vesicle, the prostate, and then the bulbourethral gland. The seminal vesicle here in the back, it produces fluids that's going to help neutralize vaginal acid as well as the acid in the male urethra. So the fluid is going to be alkaline and it's viscous, meaning it's thick. There's lots of fructose in it, so it's going to provide nourishment to the sperm cells. There are also prostaglandins that are going to relax uterine contractions. There are coagulation proteins, so upon ejaculation, the semen needs to coagulate. One that's going to act as a cushion for ejaculated sperm cells. I mean, these are actually being propelled out of the body at a fairly rapid rate. So if you didn't have this, they would simply burst upon impact, like in the female reproductive tract, or they would simply leak out. So they need to kind of be held inside the female's body so they don't all leak out, as well as cushions so they don't just explode upon impact. Again, the largest, I guess, proportion of the seminal volume will be due to the seminal vesicles. The prostate gland is about the size of a chestnut. Hopefully you all know about what a size of a chestnut that is. It is below or inferior to the bladder. Its secretions are slightly acidic. This is mostly due to the citric acid that's there for ATP production. There are also enzymes for seminal liquefaction, or liquefaction, I should say. So just like we had coagulation proteins, after about, 15 minutes or so, 10 to 15 minutes, the semen will begin to liquefy because otherwise the sperm will be trapped. So that's what these enzymes will be for. There's also a antibiotic that can help destroy bacteria. So again, just kind of keeps parts of the reproductive tract sort of free from infections as well as keeping the sperm safe. It naturally enlarges with age, so something that people with prostates will get to look forward to as they get older. It's not cancerous necessarily, there's just general enlargement with age. Sometimes that enlargement can interfere with voiding of the bladder. Again, this is just sort of a number for you. It's going to produce the next largest quantity of the seminal fluid. The bulbal urethral gland, these are these tiny two little glands here, kind of at the base or at the bulb of the penis. That's why it's called bulbourethral. It's right at the bulb of the urethra, or the bulb of the penis by the urethra. They're also producing an alkaline mucus. This is there to neutralize primarily the urine in the male urethra, as well as lubricate the lining of it. You want the pathway out of the male body as smooth and as non-acidic as possible. The best way I can describe this to you is imagine trying to slide down a hot slide without like water on it, like at a water slide. You would get like, you know how you'd rub your skin off, right? Same idea, like you need to have this secretion to produce a nice slippery surface so that the seminal fluid and the sperm can leave the body and they're not destroyed as they're trying to pass through the male urethra. So some more semen statistics for you. It's slightly alkaline, somewhat milky in appearance, and it is sticky thanks to those coagulation factors. Your typical volume as well as sperm count. So I suppose at the low end, we're talking maybe 125 million sperm. And at the high end, what is that, like 750 sperm in one ejaculate. It coagulates within 5 minutes. It again reliquefies in about 15. And then you'll see, for males, like if a couple is having difficulty conceiving, they may do a fertility analysis. If sperm do not have enough motility, like they're not swimming fast enough, or if there's a low count, and we'll see later on, or an abnormal shape, this can negatively impact fertility. All right. So this lecture will be a little bit longer than our usual ones, but rather than stopping, I'm just going to continue on because we only have a few more slides. As far as the anatomy of the penis goes, we're going to have Again, here's a urethra, and this time it's laid out kind of in this frontal plane. You can kind of see how the section is. Here's a bladder, again, prostatic urethra. We're going to have a membranous urethra, and then it's either called the spongy urethra or the penile urethra as it goes through the penis out the body. There are two main tissues in the penis. The first is going to be this much larger corpora cavernosa. This is the main erectile tissue in the penis, so this will become engorged with blood, giving the typical erection. And then the corpus spongiosum is a tissue that wraps around the urethra. It is called spongiosum because it actually looks just like a sponge. And it is there to make sure that the urethra stays open upon erection. Now, erection and ejaculation, these are going to be a delicate balance of the parasympathetic and sympathetic nervous systems. So parasympathetic, rest and digest. Sympathetic is your fight or flight, right? So this is why there's a lot of things that can go wrong with like erectile dysfunction or difficulty maintaining an erection for males, or even achieving an erection. So again, to achieve one, it's parasympathetic nervous system. So if the individual is too stressed, it may be very difficult to achieve an erection because, again, that is a different nervous system that is predominant in that case. But basically, upon sexual stimulation, the arteries that are supplying the penis release nitric oxide. And if you remember, nitrous oxide causes vasodilation. So with vasodilation, more blood enters into the corpus cavernosum, causing an erection. The veins become compressed, the blood is trapped, and again, that's how an erection is achieved. Now, there has to be a quick interchange between the parasympathetic and sympathetic nervous systems. If the sympathetic nervous system activates too soon, you may have premature ejaculation, or if the parasympathetic nervous system is inactivated too soon, it may cause the loss of an erection. But an ejaculation is basically a quick switch to the sympathetic nervous system, which causes then the muscle contractions to close off the bladder, because again, we do not want urine mixing in with the seminal fluid. That can damage and destroy sperm. Then we have muscle contractions in the ductus deferens, the seminal vesicles, all of those different organs that we just talked about that will cause the release of secretions and propel the semen through the urethra and out the male body. All right, I will stop there, and then the next thing we'll talk about is a shorter discussion about what can go wrong.